applied medical science with honor at King Saud University in Riyadh. And she received her PhD in immunology from the Department of Pathology at Dalhousie University in Canada. Dr. Jawdat is currently the director of the Saudi Stem Cell Donor Registry and Cord Blood Bank at King Abdullah International Medical Research Center. She is a senior research scientist in the field of immunology. She published vast number of publications and participation in the scientific community. Dunia is actually the chair of the scientific committee for the ARSHI, Arabian Society for Histocompatibility and Immunogenetics, and also she is serving as a board member for the World Marrow Donor Associations. Dunia also, she is active member in other societies, including in the International Society for Cellular Therapy, the American Society for Histocompatibility and Immunogenetics. Dunia is also a fact inspector that's certified by Foundation of Accreditation of Cellular Immunology, Cellular Therapy. Without further delay, Dr. Dunia today now will give us a talk about humoral immunity. Without further delay, Dunia, the mic is yours.
Thank you, Fadi, for this nice introduction, and thank you, Prof. Ali, for inviting me. I think I'm gonna turn maybe the video off for the internet. Maybe it's better, and then I'll continue. Okay. So I'm going to talk about, thank you, Dr. Nada and Dr. Omar, you made my life easier because of the nice introduction that you have given. And as you will see now, many things will be given at a different presentation, sharing between me and my colleagues, because the immune system everything is very interactive. Okay, let me just, okay. So just take you back again one step, just to make sure that you have all digest this information. Our immune response is two types, innate and adaptive. Our innate immune response is the first line of defense, and it is, we cannot say it's 100% non-specific, but it has more like a global kind of recognition. And the cells that involved in the innate immune response are the macrophages, neutrophils, and monocytes, and others, and also some molecules like complement. And Dr. Amanasa will talk about the complement later on. Our adaptive immune response is, I call them more like a SWAT team, like very specialized cells that kick off later on. It takes, for example, if an antigen enters the body, immediately it will be spot and try to be fought by the innate immune response. But the adaptive immune response, within that day, they try to become activated, but it requires time. So it's usually between 3 to 4 to 7 days until the adaptive immune response kick in. And that's why sometimes you feel that you're sick in the beginning when you have an infection, but later after 3-4 days, you feel that you're going to a recovery stage. And that's because the adaptive immune response kicks in. And it's mediated by our T and B lymphocytes. Okay, so here you just have the figure where you have a microbe. If you can look here at the very left side. So when a microbe enters an individual's body, it will be captured, or the innate immune response cells will try to get rid of that pathogen. And at the same time, parallel, you will have the activation or start of the adaptive immune response. So B cells and T cells taking care of that pathogen. So for the adaptive immune response, as we said, it is mediated by T and B lymphocyte, but also this is, we can categorize it into different types. So we call anything that is mediated by a B lymphocyte, we call it a humoral immunity. And humoral comes from the name humoral. Immunity comes from the word humoral, which is the extracellular fluid. And oops, that jumped. Sorry. Okay. And if we say humoral immunity is the aspect of immunity that mediates that is mediated by macromolecules found in extracellular fluid, including antibodies that are secreted from B cells, but also complement component and antimicrobial peptides. And we're going to focus in this lecture mainly about the antibodies from B lymphocytes. So B lymphocyte or B cells, both are correct. There are subtypes of white blood cells, as Dr. Omar explained to you earlier. So they originate from stem cells in the bone marrow, and during that time where in the bone marrow, they undergo several stages. So I'm not going to go into any details here, but they undergo stages where they also form the receptor that is specific for binding you have from 0 to 9 numbers on your screen, but there are so many different phone numbers you can call. So that's very similar. So you can have, if you only look at the first 3 numbers, you can have 1, 2, 3, 3, 2, 1, 1, 1, 2, 2, 2, 2. So depending how these genes combine together, every time you have a different receptor. That's why a kind of stage of education. So recognized cell protein need to undergo, we call it apoptosis, which is kind of like cells die within the bone marrow. That site, it's around, I think, 10% only that of B cell that actually leave the bone marrow. Okay, so what is the antibody, or we call it immunoglobulin? Both are correct. Antibodies or immunoglobulin are glycoproteins, and they're called that keep switching. Sorry. Okay. I hope it doesn't keep flipping. Okay, so they are called portion that they represent in the serum of the blood. And the main function, or the main idea about antibody recognition of antigen. And cell has a single specificity for one antigen, or more specifically for one epitope, so part of the antigen that it can bind to. So if we have millions of B cells in our blood, each B cell has a unique antigen recognition. When all immunoglobulin, they share certain structure, features by binding antigen and have effector function, and we're gonna talk about this in a few seconds. So first, I would like to take you through the antibody structure. So for those who don't know, the antibody has 2 light chains and 2 heavy chains. The light chains are either, they're called kappa or lambda, and the light chain, we have different classes. I'm gonna come to that, but what I want to know is that, as you can see, the antibody looks like a Y shape. So the for antibodies. The very part is what is important, that will bind antigen. Skipped. I hope no sound. Can you hear me now?
Yes, yes.
Hope it's clear.
Yes, we hear.
Dr. Dunia, are you using your
Yeah, you hear me?
laptop
Okay.
or phone? Dr. Dunia?
Fadi.
Yes, Doctor. Doctor, are you using your laptop or your phone mic? Hello?
I'll tell.
Oh, now is interrupted is a little bit. Try, go ahead.
Immunoglobulin classes. We have IgG, IgM, IgA, IgE, and IgD, and these are the 5 classes. It's gamma, mu, alpha, epsilon. Now, of course, they differ in size, charge, amino acid, and composition and carbohydrate content, but the function is the same, which is binding to antigen. So with IgG, for example, is the most abundant antibody, and it consists of 75 to 80%. Now, there are different subclasses of IgG. And the nice thing about IgG is that also they are and that will be covered by Dr. Amr Nasser. Also, they mediate opsonization. Opsonization, I'd like to take you through part which is variable, will bind to the antigen, but the constant region of the antibody can bind to other about the antibody. So for example, if there is an antigen and that cannot bind or cannot be phagocytosed, for example, now the phagocytic cell to engulf the whole antigen with the antibody, and is our IgM, and it is less abundant as the IgG.
Come in, go on, come in. Your voice, go on, come in, please.
Yeah, I cannot do anything about this. Fadi.
Yes, I hear you. Are you using your laptop or your phone?
His voice is lagging. His voice is lagging. Go on, come in, go on, come in, please.
Yeah.
Go ahead, Doctor. Go ahead, Doctor.
So IgM, when it's expressed, it's usually expressed as a membrane-bound and usually they like to bind together. You don't see an IgM by itself. There's always like 5 bound together, and they play an important role in the primary immune response, and I will come to that part later on. IgA is between 15 and 20%. The beauty about IgA is not they're only present in blood, but they can also be found in secretions like saliva, tears, and breast milk. And that's why we tell all our birth-giving mothers to breastfeed their children, because IgA can be transferred from the milk to the child. So they play also an important role, I guess.
We cannot hear you, Dr. Dunia.
Dr. Dunia?
They like to bond, and they're usually present, as you heard also from Neda, that they are present on mast cells and basophils. So IgA, when they are released, they're bind to specific receptor. We call them Fc receptor, bound on the mast cell and basophil allergy, because IgE, it will activate the mast cell, and then the release, of course, of their mediators would include histamine. Not a clear role, but we have seen when they are mature and leave the bone marrow. But in the blood, they consist a very, very low percentage. Function of the antibody, and I've already mentioned the main thing is binding to the antibody killers, but death of the antigen. The region of the antibody is what mediate the effect. So what are these effector functions? Activation other immune cells. So opsonization, I mentioned, I already described it. Neutralization is when they activation of other immune cells, like it binds to the pathogen, and from the constant region activate the natural killer cells to release toxic pathogen. So what are the phases of the humoral immune response? First, we have the antigen recognition, and this is for T cell activation, but they also bind to. So the B cell is antigen presenting cell. So B cell can bind directly to the antigen and then internalize the antigen with MHC, and I will come to that later on. But at this stage, they present the antigen with T helper cells. So when T helper can give help to that B cell and initiate the B cell activation by giving two signals to the B cell. The next, there's one thing that I kind of want to mention, and sometimes require help from T cells. Sometimes T cells do not require help, and they can. We call that independent antigen activation. So the T cell first becomes activated by dendritic cell, will allow the B cell activation. A T cell independent T help, and they can, and you would think that, okay, this might be better, but it's not, and I will explain to you in a second why. So one, we copy paste from that gut signals and gut cell proliferation, we have differentiation. So they do plasma cell. So the plasma cell is the cell that will release antibodies at that stage, and the memory cell will release antibody later on in the future. So we have isotype switching, somatic hypermutation, and differentiation into plasma cells. So isotype switching is for the B cell to make a different antibody. So we start with IgM, but IgA, IgE based on and enter the host. It allows the B cell to switch in binding site. We call this hypermutation affinity maturation. So the receptor slightly changes, and it could be to undergo apoptosis. If there is okay, and then response has no isotype and false. I'm trying to see. Yeah, so I think that was one more slide here. So if you have a primary immune response versus a secondary immune response, primary is the first time you have an immune response where your body sees the antigen for the first time. So it takes time, it takes a few days.
Dr. Dunia.
Response can, and it can't fail. It's either too much or too little. And if it's too much, and we call this hypersensitive, like what we see with immune deficiencies. And that's it.
Dr. Dunia, thank you so much. And I believe we have a problem with the sound. Can you move somewhere where you have a better, the change in the voice tone is so big.
Dr. Dunia, are you using your mic or are you using your phone?
No, I'm using the laptop.
It should be
microphone.
Oh, okay. Strange. Anyway, without further delay, so would you continue to your next talk now, which is antigen presentation?
665.
Yes.
If you can't just the voice, I don't know if changing the place or and also try to mute any other phones if you are recording.
Can I ask Dr. Dunia, are you using a hotspot from your mobile to connect you to your laptop or direct connection to landline?
Would that be possible to, until Dunia fix her issue, to start like with Amr and then back to Dunia? Would that be possible?
And I suggest so if Dr. Amr is ready.
السلام عليكم.
مرحبا دكتور عمرو. كيف حالك؟
الحمد لله. I can start now. Let Dr. Dunia just give me permission.
Sure. Just we'll talk to Dr. Dunia first. Dr. Dunia,